work stems from an honors thesis that was done many years ago by Max Chipman at the site with me and Dr. Pierre Pufol. Both of them were at Acadia University at the time. Both have since moved on, as have I. As Jade mentioned, I was at the time the curator at Joggins. And for those of you who are not familiar with the UNESCO World Heritage Site, the next slide shows a lovely view of part of it. This used to be my primary field site, and I know Jade is probably feeling pretty lucky to have her office perched over the cliffs like I felt for so long. Probably all of you are familiar with this site, but just in case there's anyone out there who isn't, it's been on the World Heritage List since 2008, and it's been on the list because of its well-preserved tropical terrestrial environments of the late Carboniferous period, about 300 million years ago. As such, it's particularly famous for its standing trees and forests in the cliffs. The next slide will show some of these beautiful images that Joggins is particularly famous for. Again, these standing trees and the flora and fauna contained within the forests. So well known for Hylonomus lyelli, the oldest known reptile in the fossil record, pictured to the bottom right. The top right shows a beautifully preserved amphibian, Dendripodon, which I know Jade has worked on, and other beautiful vertebrate fossils, but as well invertebrates like this land snail at the bottom left, and fern fossils, footprints. So these are really iconic images of Joggins. What's less well known are the aquatic environments. So the next slide, we will go over the Joggins Formation in a little bit more detail. The extent of the UNESCO World Heritage Site is outlined here, bracketed in blue. It's about a 15-kilometer long site encompassing 5 formations from the Boss Point up to the Ragged Reef. But by far the most well-known formation is the Joggins Formation. So I've blown that up here to the right, and you can see the red boxes, the thicker red boxes. These are the exact feces that we have been studying for this work. These are the aquatic feces. In the next slide, we'll talk about that open water feces association represented by these limestones pictured here. And what's found within the limestones are a number of invertebrates. If you just click the next, yeah, thank you, are a number of invertebrates, including bivalves, ostracods, tube worms, and crustaceans, and vertebrates, including amphibians, and quite a diverse fish community, both body fossils and trace fossils. And of course, it's the trace fossils that we're focusing on for this work. This open water feces association has been interpreted as a brackish bay. And so if you just click the next slide, that's a modern equivalent of this brackish bay environments would be things like the Mississippi Delta or Amazon Basin, where rivers are meeting the sea. So this estuarine type environment. And the next slide.
Okay, so I will take over now and to just talk about what exactly is a coprolite. And as Jade mentioned, it's basically fossilized poop or fossilized fecal matter. The word coprolite was first coined in 1829 by Reverend William Buckland, and it's from the Greek words kopros, which means dung, and lithos, which means stone. So it literally means dung stone. Not very imaginative, but it's a lot better than saying fossilized poop all the time. And coprolites are, as Melissa just said, they are trace fossils. And this is different from a skeletal fossil, which you might think of as like a shell or a bone, but they don't exactly tell you what organism necessarily produced it. You know, everybody and everything poops, so we can't say what made specific coprolites, but they do tell us a bit about the behavior of the animal. And in this case, it's specific to what the animals ate. So by looking at coprolites and looking at their contents, we can get an idea of what organisms were present in the environment and their relationships to one another. So if you look at a coprolite and you get pollen in it, it tells you about what plants were in the environment. The plankton can tell you what about if it's in the water, then what plankton organisms were there. If there's bones in it and you can actually identify the bones, then you can see what the organism that made the coprolite was preying on. So it gives us an idea about these ancient ecosystems and how they relate to each other. And this especially, as Melissa just said, in the Joggins Formation, we don't know a lot about the aquatic environment. It's a terrestrial environment that gets all the press at Joggins because it is absolutely amazing with the trees. So it's the aquatic environment that we still have a lot of questions about. And that's what really drove our interest in this, is what can we look at with these to try and figure out what does it tell us about the environment. And the other thing with coprolites too, is that they're not always preserved, which is probably a good thing, because going back to the everybody and everything poops thing, if everything was preserved, we would be up to our elbows in coprolites, or modern day equivalents, and I'm going to leave that to you guys to imagine that. So it is really good that coprolites or poop in general breaks down, but on occasion it does get preserved. And when it does get preserved, it tells us a bit about the environment which is preserved. This is especially important in the aquatic environment, because when a coprolite becomes lithified, it records the environment it was in. So if we study it geochemically, we can get an idea of the water that it was actually deposited. Was it high in oxygen? Was it low in oxygen? And things like that, that gives us more clues into the actual mysterious environments. And the other thing is the actual composition of the coprolite puts a bias on whether they get preserved or not. Carnivorous coprolites fossilize more readily than something that was made by a herbivore. And this is because in a carnivore, you're eating other animals, and animal tissues are high in phosphate. This means that the coprolite, or I guess at this point, the poop is high in phosphate and therefore easily mineralized, and then becomes easier to preserve. So you get a bias in the fossil record towards carnivorous coprolites. And a small tangent, but fun fact to the side, that coprolites are so rich in phosphate that in between late 1840s and 1909, they were actually mined in England for fertilizer. So over 2 million tons of coprolites were mined between 1849 and 1909, just in Cambridgeshire alone. And they were sent out from the docks at Ipswich. And there's actually a street there that's still called Coprolite Street. So if anyone has the desire to, you can actually go there and find where these coprolites were mined, where they were ground up, and where they were shipped out for fertilizer. It actually was, again, came back in like for World War II, again for fertilizer as well. So coprolites have been along basically as long as organisms have pooped, and it has been preserved. And they do come in a variety of shapes and sizes, as you guys can imagine. Something from like a small fish, like a small goldfish, is going to look a lot different than something that may have been preserved from a T-Rex, for instance. So they're really neat. They give us a lot of information about the organisms. And this is really important too for vertebrate paleontology. I tend to consider myself more invertebrate. I don't deal much with vertebrate, but if you are lucky enough to, let's say, find a dinosaur bone, you find a T-Rex femur, and you pick it up and you're like, yes, I know it's from a T-Rex. That's great. That's really cool. But it doesn't tell you anything about the environment, whereas coprolites can give you that little bit of window into it. So at Joggins, we are dealing with almost all fish coprolites. There is a small chance that there are some that are from amphibians, but the majority of coprolites in Joggins are from the fish, and they're all in those open water, freshwater open water feces associations that Melissa was just talking about. They are light beige. They tend to be from less than a centimeter to kind of less than 10 centimeters long. So no ginormous T-Rex size ones here. And they average about 2 centimeters in size. They're composed of calcium-rich phosphate, which is known as calciapatite or phosphorapatite. And so as Melissa said, we basically are working off of Max Chipman's work for his honors thesis. And Max collected 74 samples that were ex situ, so they were lying on the beach. And he used hand samples and took some thin sections of them. And again, it just depends if you could actually get a thin section, because as you can imagine, they can be kind of crumbly too, so they don't necessarily lend themselves well to thin sections. But he looked at the hand samples and whatever thin sections he could make to describe their shapes as well as their contents. And he was able to come up with 6 different morphotypes. So he was able to take these 74 coprolites and break them up into 6 groups based on shape. So from most abundant to least abundant, you have the small equine coprolites, the cylindrical here, the conical or heteropolar macrospiral, large, irregular, and a spiral or an amphiporal spiral. So these are just some pictures of some of the coprolites he looked at in different shapes. I would say if you want to go read an excellent paper, his from 2020 is really great and actually explains the criteria for each of these. In terms of composition, they have bones. Most of them have some kind of bones, or there are bones found throughout them, as well as fish scales. And fish scales you can see in the top right corner here is one. They were fairly rare. The bone fragments themselves were kind of found scattered throughout, and the type of coprolite, the morphotype, also it varied between them. So the small equine ones had the least amount of bone fragments in them, whereas the larger ones, and specifically the conical ones, tended to have more of the bone fragments. Now, unfortunately, the bones were too fragmented and too small to actually identify what fish they may have come from, but we do know they are basically fish bones. And in the pictures here, it can be very difficult to see the bones in all these pictures unless you know what you're looking for. The easiest one is the bottom middle one here, which is actually a blow up from the one on the left to it. And then, like I said, the top right is a fish scale here. The other thing that Max did was look at thin sections and to see what the composition did look like in there, to see if he could get any more insights from it. And the top two photos here do show some bone. So that's the, I don't know if you can see my cursor, but it's the long, thin, clear, in the kind of orangey, muddy coprolite matrix here. And that orange is from that phosphate composition as well. And the other thing that Max found in the thin sections are coated grains, and these are in the bottom two photos. And a coated grain happens when an organism ingests sediment or sand or dirt, anything like that. If it goes to the digestive tract where it picks up, it's coated in mucus and then picks up more grains and more sand, and becomes kind of this coated, layered grain structure in the digestive tract, and then it is passed out and preserved in the coprolite. And what this tells us is that whoever has the coated grains ingested dirt or sediment. Whether that was intentional or not, we don't know. It could be some kind of detritivore that is going through the mud trying to find the organic matter. It could be looking for something like an invertebrate worm that lives in the mud, anything like that. But what Max did describe was that just like the bones, the amount of coated grains varied between morphotype, and it was reversed to the bones. So the small equine coprolites, which had the least amount of bones, actually had the most amount of coated grains, and conversely, the larger conical coprolites, which had the most amount of bones, had the least amount of coated grains. So even just from this, you can start to think, okay, well, if the small equine coprolites had lower bones and more coated grains, maybe this was some kind of organism that was small and feeding at the sea floor and ingesting all the sediment. Max also undertook some cathodoluminescence study on some of the samples. So he took 4 samples, and the reason behind this was he wanted to check to see if there had been any alteration to the sample. So whenever you do have a fossil or sediment and it does get buried, it has a potential to undergo diagenesis. And this can end up leading to alteration of your fossils and different features within them. So it's always good to check whether the rock is still pretty much how it should have been, or if something has happened along the way that could affect your interpretation. And the cathodoluminescence just basically showed that there's been very little alteration, very little diagenesis. So what you see is what you do get in this case. It also told Max that these are deposited in a reducing condition. So in an area where there's little oxygen, which also makes sense. Again, poop tends to break down in oxygen, and if it didn't, we'd be in a lot of trouble. So it's pretty good that it does break down, and it's the case. The other thing that Max did was he did some CT scanning. And the reason behind this was to check that whether you could use CT images to look inside coprolites and see if they have some kind of features. Like can you identify bones by using CT scanning? And the real conclusion of this was not really, because bones are made of phosphoapatite and the coprolites are made of phosphoapatite. So they're very hard to distinguish it. He only did 2 samples. So in other words, this wasn't really the best technique to try and get a view into coprolites, but you know, that's great. It rules it off the list, and somebody down the road who has the same idea can find his paper and say, okay, I'm going to try a different method.
So we really knew prior to Max's work what fish species were here based on David Carpenter's research in 2015. And so he did a really nice job of establishing the fish community and what there was at the UNESCO World Heritage Site. This was, of course, based on body fossils. So he was looking at a variety of teeth, some of which are pictured here, and parts of the jaws, even rib bones and scales. So all of those are pictured to the left here. And what Max did was really to add to this work now to help us understand the food web better. And he was able to create this trophic pyramid, which was, of course, based on what we knew of what fish species are there, and now more about what they're eating. So we're really working on the top 3 sections of this trophic pyramid, starting at trophic level 3 with the smaller equant coprolites that he found, which we related to haplolepids, and then sort of grouped a number of different morphotypes together, which represent different species, from smaller shark species like xenocanthus, and medium to larger sized fish like coelacanth, and some larger shark species like the orthocanthus. And of course, at the top of this pyramid are the top predators, which in this case are the rhizodonts. So looking at both the body fossils and now with Max's work, the trace fossils, we really have much more of an idea of what existed at Joggins. If you go to the next slide, I'll relate those body fossils now to the coprolites. So again, the small equant coprolites, which are made by the haplolepids, and then these 4 different types. I only have 3 here because irregular, but then there's the conical and the cylindrical and the spiral coprolites, which we relate to some of these mid-sized or larger shark species. And then again, the really large coprolites from the largest fish, the rhizodonts. And so we were able to really get much more of a clear idea of what's happening in the aquatic environment now. And there are, if you ever have been to Joggins, and often I would tell people this, one of the reasons why we decided to work on the coprolites are because you cannot help but to step on them. They're literally everywhere, scattered all over the beach. And so it seemed like a really useful tool to help us understand more about what we're seeing. So now on the next slide, we go from this sort of dimmer black and white view of what the aquatic environment was like to now more in-depth, more color, more understanding of the sort of trophic relationships in the food web here. But we wanted to delve deeper, even deeper, and this is where Nikole and I moved along, and also Pierre moved along to a more of a deep dive into our next stage of this research. And we hypothesized that because of these different morphotypes representing different trophic levels, that most likely they have dissimilar elemental compositions because of the dietary differences. So we started to do a deeper dive into looking at what these elemental differences might comprise. So our methods for this were twofold so far. We use scanning electron microscopy, energy dispersive X-ray spectroscopy, so SEM-EDS, it's a mouthful, and Nikole will discuss the second method, but for now I'll start with the first. So the SEM-EDS was done at the digital microscopy facility at Mount Allison University with Jim Ehrman. And the purpose of this was to really look at broad trends in the elemental composition. So really trying to find what elements, what compounds are within the coprolites. And initially we did this work a few years ago with just a few samples from each trophic level. So I think we had about 10 samples. And we had some pretty interesting and promising results, but we thought, okay, we actually need to expand this work. So it kind of induced us to do more. And so we expanded the sample size to 42. Again, these are representatives of each morphotype across the trophic levels. And what we found was three main elements or compounds that were detected. So pyrite, which was pretty common, as well as barite and zinc sulfide. The pyrite and the barite were most common within the coprolites, but we also looked at the substrate surrounding the coprolites, and pyrite was actually pretty common in the substrate as well. You can see for the images on your right here, this is what you get. Scanning portions of either within the coprolite or the boundary of the coprolite in the substrate, and those peaks, those yellow peaks, are the compounds. So I can't expect you to read those, but those peaks represent these three main compounds, and there's some others as well, which I'll deal with on the next slide. The zinc sulfide was the least common we found in the coprolites and the substrate, but they tended to be more abundant in the substrate compared to the coprolites. Overall, though, we found that the sulfates and sulfites occur both within the coprolite and the substrate. So they're not mutually exclusive of each other. They might be present in one sample altogether, or they might all be absent, or a combination of each. And so, for instance, we were finding pyrite in both the coprolite often and in the substrate. And this is really difficult for us to distinguish if really are we looking at something that had been digested by the fish, or is it some environmental influence here? So, or a possible diagenic crust, because what we were finding is that often these occur at the boundaries of the coprolites. So, oh, and I also will note that there were many other elements that were found. So those peaks, those yellow peaks in the previous slide, some of them were zircon, etc. But kind of minimal. We mostly found barite, pyrite, and zinc sulfide. Overall, the next slide, I'll show you that there's no obvious correlation between the elements or compounds detected and the morphotype. So again, I don't expect you to read this, but on the columns, the rows to the left are color-coded based on morphotype. So, we might expect that within each of these samples within the morphotypes, we might find similar types of elements, but not necessarily did we find that. So we were looking at, again, the coprolite and the substrate surrounding it. And again, no obvious correlation between these elements and the morphotypes. So not looking like our hypothesis here is being supported by this method.
Okay, so the next thing we thought we would try was laser ablation ICP-MS. And this method kind of builds on the SEM work, where the SEM is a bit broader. The ICP-MS method allows us to do really detailed, really accurate elemental analysis. So this was done at the Queen's Facility for Isotope Research, so QFIR, and it was done with Pierre Poufal, who had a fourth-year student doing this as a student independent project. And the way this works is you basically take a laser and you can adjust the beam width of it, but you can get it incredibly small, and you shoot it at whatever it is you want to analyze, in this case, the coprolite, and you vaporize part of the coprolite. And this is done under vacuum. And the particles that are then kind of called ablated are transported to a chamber where they're put into a mass spectrometer, and you can measure the elemental composition of whatever it was that you just shot with the laser. This can get down to parts per billion in terms of accuracy. So it's a really good way to try and get an idea of what is in your rock. So like I said, we weren't sure if what we were seeing was diagenetic crust. Was this what it is all the way through? Because you don't get that kind of resolution with the SEM EDS. So this student, whose name is Ashley, she selected 7 of the samples. And they did a spot diameter of about 75 microns. And each sample that they did, they ablated 15 times. So they did 3 tracks on each sample and 5 spots each, spaced 300 micrometers apart. And the idea with this is they were able then to map across the coprolite in very specific regions. So you're not just dealing with something that might just be on the outskirts of the coprolite, you're actually looking at it in it, and you're also looking at it across the coprolite as well. Now, the main drawback with ICP-MS is one, it's time consuming. It takes a long time to run all of this. You have to stop and check standards all the time, and it's very costly. So the 7 samples were kind of what we were pushing with our budget and time constraints for it. And it was just an idea to try and see if this gave us any additional information as to go along with the SEM EDS. So the data that they got from it, it was for 79 channels, which represent different elements, but the main focus was on rare earth elements as well as yttrium, and to kind of see what this changes in the coprolites at all, and again, throughout the different morphotypes, if possible. So these are the samples in the ICP-MS chamber. And the other thing when you do ICP-MS is it gives you a crap ton of data. I'm just going to say, but it does give you a whole lot of data that you have to do some statistical reduction to, because it is just so much that you need to figure out what it's actually saying. So the first thing that was done was stoichiometric normalization to correct for analytical drift. So this means essentially that sometimes when you're going along and you're blading it, you have to keep checking with the standards, but instruments can be off. And if they're not set up right, or if the sample isn't perfectly level, that all can change it very quickly. So what you do is you use a standard where you have a known elemental amount. So in this case, it was done using calcium. So use a standard for calcium where they knew exactly the amount of calcium in it. And then they measured it with the machine and they compared it to figure out how much it was off. And that creates what's called a correction factor. And then that is applied to all the data to correct for anything that may be slightly off with it. The other thing is you need to figure out if what it's telling you in terms of concentration is significant or not. Because these elements technically exist in almost every rock and in our everyday life. So how do we know if the differences we're seeing are actually significant, or if that's kind of what they're supposed to be? So you normalize the REs and Y data in this case against what's called post-Archean average shale. So these are known values from the crust that have been published, and they get updated every so often. And you figure out, okay, this is what it should be, and this is what it is. So if it equals 1, by comparing them, then you know that that's sort of what you expected. If it's above 1, you know that you have an elevated concentration of it. So you think, okay, why is it then elevated? So that kind of forms the basis of the ICP-MS data. So Ashley was able to take this data and then plot it up in a few charts here to the right. And I'm just going to stipulate here that we are still working through this. So bear with us while we'll give you kind of a rundown of what we know so far. So we do have some samples, specifically the top blue and yellow ones here, that registered elevated concentration of rare earth elements and yttrium. Why the other ones didn't, we're not sure. We're still trying to figure out what's what. We also get subpeaks in a few specific elements. So we have it in yttrium, we have it in selenium, and we also have it in, just making sure I'm saying this right, it's in the gadolinium, which is the GD one here, and these are minor subpeaks here. There's also a small drop in the praseodymium here. So we did get some results, and they are curious. For instance, the one that is higher seems to be a conical one, but for us, it's also like, okay, well, that occurs at the same trophic level as some of the other ones in here that plotted lower. And what we really kind of expected to see was our small equine varieties, which have the least amount of bones and should therefore have the least kind of amount of crazy minerals in them, to be plotting outside of the main stack there, which we don't see. So our so far preliminary observations on this is that this is perhaps, just like SEM-EDS, not going to be the best method for using to determine trophic level. So what does it all mean? Put this together with the SEM, what is it? Do we see elemental differences related to morphotype and therefore diet? Well, so far both the SEM and ICP-MS haven't really given us definitive answers on that. We don't see any definitive pattern. We don't see anything that's repeating. We don't see anything, for instance, like we don't see the small equines plotting below that would explain kind of a different trophic level. We don't see this distinction between the 3 different trophic levels in our data. But that still doesn't answer the question is why do some of them plot with elevated REEs and yttrium? Like, why do we have certain ones that do have rare earth elements and yttrium? Where are they, and where are they coming from? And where's the selenium and gadolinium coming from? Is it an environmental thing? Is it specific to these coprolites in general? Is this a morphotype thing? We're not entirely sure yet. And how do we relate the ICP-MS data, if at all, to the SEM-EDS data? Right now, they both tell us that there aren't correlations between what we thought there were, and that's fine, but can we put them together to try and figure it out? So that's kind of where we are at. And the basic breakdown of this is that more research and analysis is needed to flesh out the data, totally pun intended, as well as a larger sample size, especially for the ICP-MS. We only had 7 samples, but we're running into the whole wall of budget. We need funding for this. We also need probably a dedicated student if we want to take this farther. We are also in the process of kind of looking at isotope data as well to see if that helps us. But right now, we just have a lot more questions than answers. So we're going to finish that up here. And we just want to acknowledge Max for the wonderful work that he did on his honors thesis, Pierre, Jim, Ashley, as well as Geomapping for Energy and Minerals. It's a GSC program that funded a lot of the analysis, and Joggins, Acadia, Mount Allison, and Natural Resources Canada.